We'll talk about the happenings first. What's going on? What's going on in the world of chaos, right? Here's the things that we can control, the things that aren't chaos, our class. So we're going to fill in these slides. I've asked you to open them up in their Google Classroom. You'll find them under week 17, review of information it's called. We're going to discuss a few things that you struggled with from the interactive notebook that you did on Wednesday. Checkpoints seem to be a point of struggle. Labeling the cell that was going through division, and then calculating the time spent in each phase of the cell cycle. Those were our struggling points, so we're going to do that. Outside of class, we're going to keep practicing with mitosis in the form of a gizmo. So after class, you're going to work on the gizmo, and you should have a good hour and a half to get that done. Let me know if you need your login resent to you. And then over the weekend, I'm going to ask you to pull up your exam review packet. It's, I think, 12 pages long, which seems very overwhelming. I tried to give you plenty of space to answer, so that spreads it out. But we're going to work on chapter 9, which is what chapter we're in right now. Okay, so you're reviewing information that we're talking about. We've covered everything except for the last question, which is stem cells, which we'll cover Monday. Okay, so Tuesday, I'm going to field questions over this section. So I'm not posting an answer key. You guys need to do the work yourself to be ready for your final exam. I know that I can't control final exam entirely. I know that you may have worksheets open while you're taking your test. What I can control is that they're not my worksheet. And that's one of the reasons I'm not giving you an answer key, because it would be easy enough to just open that up during exams. And that's not fair. You need to do it yourself. But what I will do is answer questions along the way. And Tuesday, I am fielding questions over chapter 9. If you don't do your review over the weekend, you won't know if you have questions, right? So that's your chance to get any blanks filled in with help, okay? So do that portion over the weekend. A reminder, back at the beginning of the week, I gave you 2 videos assigned, and I said, anytime this week, watch these videos. There's CK12. There's not a worksheet you need to do. There's no notes you need to take. It's just one of your helpful sites that you can get information from, okay? So after you watch the video, CK12 will mark it done, and I'll be able to move you from this assigned category into this graded category. So I see the majority of us have done that. Kudos to us. Last hour, about half of them had done it. So you guys are ahead of them. But I need these 6 individuals to still complete that. It's due at 3 today. Oh, I did not change your due time. I'll change that. Okay. Are you guys ready to move forward? Any questions over the agenda or the goings-ons? Shout it out, you guys. I often miss my chat box, so I'll try to pay attention to that. But you guys can help me out. So here's the first thing I saw that we struggled with. I had written the answer in, but I guess I deleted it when I changed from 4th to 5th hour. So I couldn't actually find the slide when I wanted to find it this morning, but I know that I saw it when I was answering your questions over yesterday's work or Wednesday's work. So it asked what a checkpoint is, and it wanted you to gather that information from a worksheet or from a graphic. So basically, I need to get out a presentation. So basically, checkpoints are stop and go signals. I think of this as the quality control of our cell. So you can see there are 3 checkpoints. There are 3 checkpoints during the cell cycle. So basically, it's checking to make sure everything that needs to happen has happened And if it has, chemical signals allow the cell to continue its cycle. If it hasn't happened, the cell will exit the cell cycle and be decomposed basically, or it'll go through a repair process to fix those errors. Okay, so there's 3 checkpoints. The first checkpoint is at G1. Remember, this is when the majority of the cell's growth is occurring. Has the cell gotten big enough? Is the environment favorable? And have we replicated like all of the organelles we need? Do we have enough material basically to support 2 new cells? If so, the cell continues the cycle. If not, it either repairs those errors or it kicks the cell out of the cycle and a process called apoptosis occurs. Apoptosis is basically your cells, your other cells, degrading this cell. Kind of like white blood cells will come and eat it up. The second checkpoint is at the end of G2. Have we replicated the DNA? Is there a copy of every single one of my chromosomes? And are they without error? If so, the cell proceeds. If not, it kicks it out and destroys the cell, apoptosis. The next phase is during metaphase or mitosis. Are all of the chromosomes lined up at the equator and ready to divide? Again, if not, it either corrects the error or it destroys the cell. So your body is fighting cancer every day. You're not aware of it because your body is doing its job, right? If we miss any of these checkpoints, that's when cancer occurs. So you may be predisposed to cancer, in which case the genes that control these checkpoints may malfunction, okay? But our cell is fighting it by correcting the errors or destroying the affected cell. So we don't even know it. We don't know that we're fighting cancer because we're winning. We're doing the job we need to do. The next page that I found people had errors on was labeling of this cell cycle. And I don't know if it's because there's a lot of words there, and so we just skipped it because it seems like a lot of work, or if we really didn't know what to do, okay? So first of all, notice the capital letters A, B, C, D, E, F. That's where we're going to put the phases of the cell cycle. So remember, I, PMAT. That's your mnemonic. Interphase, prophase, metaphase, anaphase, telophase. I, PMAT. Which cell do you think is the first cell in the cycle? Where is my interphase? A, B, C, D, E. Shout it out or put it in the chat. Which one is the first cell in the cycle? A, B, C, D, E.
E.
So I'm getting a lot of answers in the chat box. Oops. So we want, notice we have a nucleus still here at E. Yeah, there's a nucleus here, there's not a nucleus here. Like the chromosomes have burst out of that nucleus and they're all over the place. So this one is going to be our interphase. I want to make my words look different because I do think it was difficult to tell between. Ooh, okay. So if I go around the clock, then there's prophase at where my chromosomes are in no particular order, right? No particular order. I do see chromosomes here, whereas in interphase, I don't see chromosomes. I, PMAT, I might have wrote that at the bottom of your page. Meaning interphase, prophase, metaphase, anaphase, telophase. The key is figuring out where it starts. Metaphase, they're all lined up at the equator. So chromosomes are in a line. Anaphase, I have 2 lines. they're being pulled apart. The chromosomes are being pulled apart. And telophase, the cell is beginning to split. Perhaps cytokinesis happens during this time. I have two new nuclei forming. I should have made them different colors because I do think all of the red kind of split. I tried. Okay, so then we want to label what is inside of the cell. So number 6, this is the outside of the cell, right? So what surrounds the outside of the cell? Chapter 7, section 7.2. What kind of membrane is outside the cell? Is it nuclear? Is it plasma? Is it cytoplasmic? I'm just going to call it the plasma membrane. Yep. Okay, and then inside of the nucleus, these letters are kind of hard to, looks like I'm on the, I think I'm here. So this little thing here that looks like a fly, that's our centriole. That's where the chromosomes or the spindle fibers come from, centriole. So if you didn't fill this out, or if I said see slide 6 or whatever, you guys can, after we do this together, you can go back and fix yours on your interactive notebook. In the middle, this is the nucleolus, little nucleus. And what we see inside is the genetic material, chromatin, chromosomes, or chromatids at this point. They're chromatin. So, 1A. Thin thread-like form of DNA, you can't see any individual chromosomes, right? Whereas here, I can see individual chromosomes. Here, I can see individual chromosomes. Here, they're being pulled apart. These are your sister chromatids. Since it's pointing at both of them, it's referring to that reference point, okay? So that's cell 1. Prophase, what do I see? I see individual chromosomes. So chromosomes, sister chromatids join together to form chromosomes. The nuclear membrane is starting to dissolve. This one you might not have known. The spindle fibers start radiating outward from what's called an aster. I didn't use that word this year, I don't think. And coming from the asters, this one is trying to point out the spindle fibers. These individual lines are the spindle fibers. They're coming out. They're going to connect to chromosomes. They're what move the chromosomes around. And in the end, we form 2 new daughter cells. The key, remember, each have the exact same number of chromosomes as the original parent cell. So on the last slide, it said humans have 46. After mitosis, all our cells have 46. Said the dog has 72. After mitosis, all of the cells would have 72. Some of us were cutting that number in half. That is one of your exam questions, where I give you a chromosome number and ask you how many will be in the daughter cells. It'll be the exact same as the parent cell, okay? Any questions on this slide? Okay, so the next thing we struggled with was this table. So some of us left it blank. Again, I don't know if that means we just looked at it and were flabbergasted and it's too much information and so we didn't try, or if we just found it very difficult and we needed some help. So we're going to slow it down and back it up. So let's look at this drawing, okay? Take a minute and see if you can count up. There's 18 total cells. How many cells are in interphase? Take a minute to fill in your own answers here. How many of the 18 are in interphase? In interphase, there is, I'll write my key IP map. In interphase, I can see a nucleus, a nucleolus, no individual chromosomes. Count up how many prophase do you see? Prophase is the nucleus is starting to disappear. Chromosomes are visible. Prophase lasts a long time. You can find them in different versions, whether the nucleus is there, maybe it's not there. During metaphase, chromosomes are lined up at the equator, a single line. Write that number. Anaphase, I see chromosomes being pulled apart. Telophase, I see two nuclei forming. Okay, so interphase, circles like this, nucleus, nucleolus, visible. Ellie, how many interphase did you see?
I counted 9.
Me too. Okay, prophase. Prophase is a tricky one because it can look lots of different ways, really. The nucleus is starting to disappear. It may be in all different versions. The nucleolus chromosomes are starting to condense and become visible. Lots of different stages. Prophase, like this. How many prophase did you find? Braden, how many prophase did you find?
Isn't there just 2?
There is, yep. I found this cell number 2 and number 14. Metaphase, they're lined up at the equator like this one, a single line. Isabella, how many did you see?
I found 3.
That is correct, good work. Anaphase, there's 2 lines of chromosomes like this one here. They're being pulled apart. Evan, how many did you see that look like number 7? Evan, how many anaphase did you count? You can't hear me? I can't hear you. I'll go to Abby. Abby, how many anaphase did you count?
2.
Yeah, I just had 2, 9, and 7. And then telophase, looks like I accidentally deleted my telophase. Telophase kind of looks like you have twin cells, like this one here, number 3. How many, let's try Elizabeth. How many telophase did you count?
2.
Yes, okay, good job. Okay, so those were easy, much easier than the picture I gave you, right? This picture was kind of rough. So let's go back to the drawings. The next thing you had to do was calculate the amount of time the cells spent in each phase, okay? So we're going to calculate the percent of the cell's life in each phase. Basically, you just take the number that you counted and divide it by the total number of cells. So 9 divided by 18 is what? 50%, right? That was an easy one. 9 divided by 18. Can you take a second and calculate the percentage for prophase through telophase? take a moment to calculate prophase through telophase. So we're all filling this in together. And I'm going to call Ethan, are you with us? All right. Jonathan, what percent did you come up with for prophase?
I got 11%.
Okay. And since 2 other phases are 2 also, can I just skip ahead and say both of these are going to both be 11% as well? Because they each had 2 cells, 2 divided by 18 is 11. So metaphase, who can give me metaphase? Sophia, can you tell me what percent of the life cycle the cell spends in metaphase?
Yeah, hold on. Let me- I think I got about roughly 25. Maybe a little less.
Okay, I'm just gonna do it on my calculator. See, it sounds high to me. So I'm doing 3 divided by 18. Because there's 3 cells in metaphase and there's 18 total cells. And I got 16.7, so I'm gonna round that off at 17%. So divide by the total number of cells, which in this case is 18. All right, now let's say the cell spends, I didn't give you this one. Let's say the cell spends 120 minutes in to go through the entire cycle. 120 minutes to go through the entire cycle. How could I calculate the amount of minutes it spends in each phase? Anybody have an idea? Spends 120 minutes to go through the complete cycle. How could I calculate how much time is spent in interphase? Any ideas? If it spends 120 minutes in the cycle and 50% of that time is interphase, how many minutes is interphase?
60.
Yes, 50% of 120 is 60 minutes. Okay, what percent of, or how many minutes would it spend if it spends 120 in the entire cycle? How many minutes would be in prophase? Eleven percent of the cycle is spent in prophase. So you would say, what is 11% of 120?
I got 13.2.
Yes, me too. Oops. And since it also spends 11% of its time in anaphase, I can assume 13.2 minutes spent in anaphase and telophase as well. That leaves metaphase. 17% of the cell cycle is spent in metaphase. If it spends 120 minutes in its entire cycle, if it takes 120 minutes to go through the cycle and 17% of that time is spent in metaphase, How much time is spent in metaphase?
I got 20.4.
Love it. Thank you guys for shouting out. 20.4, correct. Okay, so that was another task that was asked of you to complete this row here, this table that you guys had, okay? So we're gonna do just this one row. Oops, let's do this one too. And we're gonna do the time spent. And where we were doing 120 minutes, notice it's using 720 minutes. So it has a different total time for the cycle. And we also need to calculate the percent of the cell cycle. So notice when we did the last slide, we said number of cells in phase, and we divided that, like how many in metaphase, and we divided that by total number of cells. Right? That gave us the percentage, multiply it by 100 to make it a percent. Okay. So you guys had a picture that looks like this, and to be honest, I should have given you different pictures. I was taking a shortcut because I was caught up in all the news Wednesday. I took a shortcut, and I guess this wasn't because of the news, because that came later. But anyways, I gave you pictures I used in AP, and I could have picked easier pictures. I could have picked pictures that looked like this, and that's what I should have given you. So for the sake of this activity, we're going to use this picture because it's easier to identify than this picture. Okay, so take a minute, count up how many cells look like this, or this, or this. So this cell is in interphase. How many cells look like this cell? You can record that on this green row. This one here, this is in prophase. Prophase has a nucleus that's beginning to break apart. I'm starting to see thicker chromosomes. How many do you see look like prophase? Write that number down. Chromosomes lined up dead in the center, that's going to be metaphase. How many do you see that look like metaphase? Count those up. Write them on the data table. Anaphase. Chromosomes are starting to be pulled apart in various extent. How many do you count to be anaphase? Oops. And finally, telophase is kind of like an elongated cell. Looks like it has two nuclei. It may or may not have the beginnings of a cell plate. How many do you count that look like a cell with two nuclei? Write that down. Originally, we didn't count those that we couldn't see. Like over here, it told you not to count the ones that were not really in focus. It's hard to tell. Okay, so like on this, maybe you didn't count, you might not have counted these cells here, because maybe they were hard to see. Maybe you didn't count these here because they were hard to see. So let's go to another page to get more people. Elise, how many did you count in interphase?
15.
15, okay. I counted a higher number, but then I maybe saw what we considered blurry, we might have disagreed with. Okay. Janet's gone, sorry. Okay. So I counted 38. Did anybody count a number like that high, or were you more down at 15? I'm going to use 15 because that's what Elise counted. Prophase, how many did you count for prophase? So I counted 7. Metaphase.
4.
Okay. So 1, 2, 3 up here I saw. And I did not see a 4th one, but if you don't have your glasses on, one of them maybe look like that. Okay. So I'm going to put 4 because that's what you saw, but no, I'm going to put 3. And then let's see, Kylie, can you tell me how many you saw that were anaphase? Anaphase, you're looking at ones that are being pulled apart. I got 6 as well. And then telophase, let's go to Ethan, I've already called on you. Are you here? Malcolm?
I'm
Okay.
just vibing.
You're what?
I'm just vibing in the call. You want me to count something?
Yeah.
All right.
That's-
Which
that's-
one?
Telophase, please.
Telophase?
Telophase.
There's about 2 that's telophased, but
Good job. Am I on the right page now? Okay. So telophase is 2. I think I wrote on the wrong slide. 6. Okay. So when I counted up how many we could see, we could only see 56. Okay. So the next column, I think it's easiest to fill out your percentage before you do the time. Okay. So the percentage, how many cells you saw in a certain phase divided by the total number of cells, just like we did here. Okay. So in interphase, there were 15, and we're dividing that by 56. So everybody take a minute to do the math. What columns do we put the answers in? This column, this is percent of cell cycle, so it's yellow. For me, I find it easier to figure out the percentage before I figure out the time. So the number that we counted for each, I should have gave you guys a new number because, did you use the 56 like I said? Jonathan, did you use 56 for your dividing number? Yeah, that's because I forgot to change because I originally had used 38, but that's okay. Okay, so for all of these, we said to divide by 56 to get the percent the cells spent in each phase. So 15 divided by 56, 7 divided by 56. All of this same, 6, 6, 6, 6. Okay. So if you went 15 divided by 56, I got 26. Did you guys get 26? 0.26%? I got 0.13. So 13%. I got 0.05, so 5%. Got 0.11, so 11%. And 0.04, so no, yeah, so 4%. Then to calculate the amount of time spent in each cycle, we need to divide by 700 and, no, not divide by, we're going to multiply by 720 minutes. Put it in our decimal form for it to be a percentage, right? So multiply each of your percentages by 720 to find out how many minutes it spent in each phase. 720 times 0.26, for example. It spent 187 minutes in interphase. So take a minute to do all of those calculations. Either one, you can keep one decimal if you want, or you can round it off to the nearest whole number. I have 13% times 720, 0.13 times 720. Did you get 93.6%? Just hopped in on some of the student work and I didn't see that number, so I wanted to double check. If you did 5%, that's 0.05 times 720, because we're using the 720 as our time, the amount of time for the full cycle, you would get 36 minutes. 11%, 0.11 times 720, 798. 79.7. 4.04 times 720, 28 point, if I did my math correctly. These aren't going to add up to 100 because I should not have been using 56 in the first place because we used a different number for interface. So we actually would have done 10, 18, 28, 33. Should have used 33 right here. That would have been correct. So then this would have added up to 720. So this is the slide you guys had. This slide was more difficult. Okay. Looking for cells in interphase. So that would be interphase. This would be interphase. This would be interphase. Looking at cells in prophase where they're starting to break apart. Let's see, a prophase. This looks like a good prophase. This is a prophase. This is a prophase. So the nucleus is starting to break apart. Doesn't look perfect anymore. Metaphase, where they're lined up at the equator. Not sure I saw any metaphase in this picture, actually. I do see an anaphase. Anaphase, they're being pulled apart here. It might have been the only one I saw on this particular slide. And telophase, let's get a telophase. I see a telophase jumping out at me right here. Maybe this is a late telophase up here. Maybe. So the slides you had to be fair. These were hard slides to analyze, but as long as you counted and you did your math, you'll get where you need to go. And if you struggled with that slide, you could go back and fix it now. Or if you haven't done your slides yet, you hopefully understand how to do the math. Any questions on the math for filling this in? Okay, so the last thing we have, you can, now anything we've covered with mitosis, cell cycle, I tried to hit the parts that I knew were struggling as a big group. You might have pieces that you were struggling with that I didn't identify, because, so you can either shout it out or put it in the chat box. This is your time to get help. Or you can write me a little private note in this slide and I'll read it over the weekend and I'll try to help you out that way. Okay? Going back to the agenda. We got the slides filled in, turned in. Today you're going to do more practice using the gizmo. And over the weekend you're going to work on your chapter 9 portion of the exam review. We're not going to have a separate chapter 9 test. We'll just have what's on the exam. Any questions of the goings-ons or the topic? Okay, so you can stick around if you want individual help or you want to talk about the world or you want to talk about yourself, anything. Otherwise, you guys are free to go. Thanks for coming.